Hello and welcome to this video. In this video, I'm going to go over some basics on how gene expression is regulated in prokaryotes. We're going to start with an overview about what gene expression means and how it's changed and why you would want to regulate gene expression, and then cover a few examples of how this occurs in prokaryotes such as bacteria. This includes regulating transcription through the use of sigma factors. A very important example in how the Lac Operon, specifically a certain set of genes, is regulated in some bacteria, and then a few other examples, other operons, for example the Trp biosynthesis operon, and some examples of post-transcriptional control. A lot of the behaviors of a cell, its ability to respond to its environment, a lot of the sophisticated cell behaviors depend on changing the gene expression, or which genes are expressed, how much they're expressed, and when they're expressed in the cell. Right? So this often means, for a cell, changing the amount of mRNA and ultimately changing the amount of functional protein, either functional RNAs or functional protein, folded proteins that can carry out some function such as an enzyme or a protein transporter. Right? Having those in the cell active will produce certain cellular behaviors or properties. So let's look at a couple examples. For example, we can regulate the amount of transcription that occurs in prokaryotes. So at any given gene, whether or not that gene will be transcribed. In some prokaryotes, RNA polymerase recognizes promoters through the help of another protein factor known as sigma factor. And in this case, different sigma factors will recognize promoters that have slightly different sequences. This leads to having sigma factors to respond, different types of sigma factors to turn on genes in response to different types of environmental stress, for example. There will be a normal sigma factor that will express genes that are important during ideal conditions. There may be a sigma factor that will only be active in conditions of high heat, and this will activate genes that are necessary for the bacteria or the prokaryote to adapt to those conditions. If you recall, RNA polymerase is a multi-subunit enzyme. Here's a couple depictions of a prokaryotic RNA polymerase, and shown in dark blue is the sigma factor component, which is actually only important for initiation of transcription. You can see here that the sigma factor is actually what allows the RNA polymerase to properly recognize a promoter. So for example, we can have RNA polymerase in a prokaryote such as E. coli, and it can have sigma 70, a version of sigma factor associated with it. This will allow it to transcribe any genes that sigma factor can recognize as a appropriate promoter, and genes that have a promoter not recognized by sigma 70 will not be transcribed. We'll produce an mRNA, and then through translation we'll produce functional protein. So here, out of these different genes, we are activating only some of them. At a higher temperature, RNA polymerase will switch, for reasons that we'll see later, will switch to use a different sigma factor, in this case sigma 32. Now, RNA polymerase with the heat shock or heat stress sigma factor will not be able to recognize promoters as it did before, but now recognize a different set of promoters, producing a different set of proteins, which again, probably going to be important for a bacteria's response to high temperatures. For a lot of genes in prokaryotes, they're arranged in an operon. And in this case, we have a single mRNA, transcription of which starts at a single promoter, but will actually contain multiple genes or multiple coding sequences. There will be several start and stop codons representing several different proteins. This type of organization of different genes makes sense, especially if the genes are all involved in the same activity. For example, these could all be genes that form enzymes in a metabolic pathway. it wouldn't make sense to make only one of these genes because you wouldn't really be able to complete the entire pathway. It only makes sense to, if you want to actually carry out that metabolic pathway, to make all of those genes and to make them all in the same relative proportions. Therefore, having them all under the control of the same promoter means that they could all be switched on by activating just transcription of that promoter, not by activating transcription at several different promoters. Let's take a look at what this looks like. All right, we're going to take a look at a specific example on how the operon for lactose metabolism in E. coli is controlled. Lactose is a sugar that the bacteria can metabolize for energy. And we're going to see that there is a protein that helps regulate transcription of this operon and all of the genes in the operon. This protein is known as the Lac protein, and it is a repressor. So here we see a depiction of the promoter for the Lac Operon. You can see both the promoter sequence, where RNA polymerase binds, and the first gene in this operon. As we'll see in a moment, there are several other genes that are all controlled by the same promoter. The Lac repressor is a protein that will, again, as many other proteins do, fold and adopt a certain shape. But instead of taking a shape to act as an enzyme, this protein is able to bind a specific sequence of DNA, here shown as the operator. This sequence is recognized by the Lac protein, and it will bind there, and it will physically prevent RNA polymerase from continuing transcription. So as long as this repressor is active, transcription will not occur. This is what you want if there's no lactose present. If there's no lactose in the cell, it doesn't make sense to produce the genes for metabolizing lactose. You would be wasting energy doing all of that transcription and translation. When lactose arrives in the cell or shows up, or if lactose is present, then you'd want a way of removing the repressor and allowing transcription to occur, because then in that situation you do need those genes. This occurs, and this will happen if lactose is present, because the repressor will actually be unable to bind the DNA if it is also bound to lactose or a similar molecule. In this way, lactose, or in this specific example, allolactose, acts as an inhibitor of the repressor, and therefore, if the repressor no longer can bind DNA, RNA polymerase can recognize the promoter and is unimpeded while it goes to transcribe the rest of the operon. Here you can see how the operon actually contains several different genes, in this case for 3 different proteins, LacZ, LacY, and LacA. Right? Each one has a start codon and a stop codon and produces a separate protein. So far we've seen how the Lac Operon, the level of transcription of the Lac Operon, responds to levels of lactose. But that's not the only signal that's used in determining how much transcription you get at that operon. So in addition to having a repressor that blocks transcription in the absence of lactose, we also have a protein that can help activate and increase the amount of transcription at the operon, and this will be active only in the absence of glucose. So when there's low glucose in the cell, one of the consequences of this is the production of a molecule known as cyclic AMP, or CAMP. And this signal, signifying low glucose levels, can bind this CAP protein, which is an activator. And only when the low glucose signal molecule binds CAP will CAP be able to bind near the promoter. And here, instead of blocking RNA polymerase from transcribing, the CAP protein will actually activate RNA polymerase and help promote transcription. So if we have a cell that only has lactose, first of all, we know that the lactose will make the repressor inactive. There's very low glucose, so the activator will be able to bind the DNA. So this should give us basically the maximum amount of transcription possible from this operon. If lactose is present, but we also have glucose, then the cell might might prefer to use glucose instead of lactose. In that case, we'll have lactose, so the repressor will be inactive, but since glucose is present, the activator, the cap protein, will not be active. So now RNA polymerase can bind the promoter, it can transcribe the genes for degrading lactose. After all, lactose is in the cell. But it won't do so to quite as high levels as we saw on the previous slide. And again, this is going to be the way of the cell to prioritize the use of glucose for metabolism rather than lactose. So we see by having these two different activators and repressors with different binding sequences around the promoter, the cell is able to actually change the level of gene expression for the Lac Operon in response to two different environmental signals. So this is an example of responding to environmental cues directly by regulating transcription. The protein that's actually binding the small molecule or the environmental signal is directly regulating transcription. This could also occur, not directly, but indirectly, through a series of different protein-protein interactions. And in prokaryotes, a common example of this is what's known as the two-component signaling system. It's essentially just one protein more complicated than the system we just saw. This also gives you a little bit of a preview of another way of gene expression or protein function changes that can occur, and that's protein modification. In a two-component signaling system in bacteria, we'll have one protein that will be a transmembrane protein. Right? It will act as a receptor for some outside signal, and it also has several other functions. We'll have another protein component here shown in purple that's going to act as the transcription factor, either the activator or the repressor. In this particular case, the activator. We also have depicted in orange, or gold, the RNA polymerase, which will be able to bind to the promoter and transcribe the gene. But right now, this is a relatively weak promoter. We're not going to get a lot of transcription. We're going to need, in this case, the transcription factor as an activator. We're going to need the activator to help RNA polymerase. But right now, the activator is inactive. And unlike the previous example, we're not going to have it bind to a small molecule. The activator itself is not going to bind to a small molecule to enable it to bind the DNA. Instead, what's going to occur is that the outside signal, this could be a small molecule, this could be light or a heat stress or some other environmental signal, will bind to the membrane receptor. This will cause a change in the protein structure for that receptor. And now we've had a change that's activated what's known as the kinase domain. And this part of the protein acts as an enzyme and is able to attach a phosphate group to one of the amino acids that makes up the transcription factor. So this is an example of a post-translational modification, or protein modification. The transcription factor protein is going to have a new molecule added to it, a new covalent bond created, and it will modify its activity. So we're going to add the phosphate group to the transcription factor, and this may change how the transcription factor functions. And here, in this crude example, I'm showing a change in shape. Now, once it has the phosphate group attached, or once it's phosphorylated, the transcription factor can bind to the promoter and help encourage RNA polymerase to bind the promoter and transcribe the gene. In this way, gene expression is increased in response to the outside signal, even though that signal is not being directly sensed by the transcription factor. There are yet other examples of how transcription is regulated in prokaryotes. This somewhat complicated example we're going to look at involves another operon, the Trp Operon. This encodes genes necessary for the biosynthesis of tryptophan, one of the 20 amino acids. And we'll see that this operon, again, has multiple levels of regulation, and one of which has to do with transcription termination. So similar to the Lac Operon, we have one promoter controlling the transcription and expression of several different genes. And we again have a repressor that can bind an operator sequence. When tryptophan is absent, the repressor does not work. And this makes sense. If tryptophan is not around, you need the genes to synthesize it. If tryptophan is present, it can bind to the repressor, and the repressor now can bind the DNA and prevent, or at least greatly decrease, the amount of transcription that occurs from that promoter. Keep in mind that this molecule will bind, but the tryptophan may become unbound, or the repressor might not always be bound to the DNA. Depends on the relative levels of the DNA and the repressor, and depends on diffusion to find it. So you may still get very small levels of transcription that occur even when tryptophan is present. This was also the case with Lac Operon. So the cell actually has another way of preventing or further decreasing the amount of transcription that occurs when tryptophan is present. Even when tryptophan is present, even when RNA polymerase will bind and still be able to, on those rare occasions, produce or begin transcription, it will also have to pass another layer of regulation that involves a transcription terminator. This is a somewhat complex example, and it actually involves coordination between transcription and translation. Before the rest of the genes in the operon, there is a small protein coding region known as Trp, and it will begin to be translated before transcription is finished. And in fact, there are signals to help coordinate the speed and the timing of both transcription and translation. Proper or improper translation of this leader sequence influences whether or not transcription will terminate. Here we can take a quick look at the beginning of the mRNA for the Trp Operon. And what's shown in different colors is the Trp sequence. What you may notice is that there are certain regions in this mRNA which can actually base pair with itself and form some secondary structures. There are also a couple important codons encoding for a Trp amino acid in the Trp protein sequence. If tryptophan is present, these codons can be decoded, the Trp protein can be produced, and the result of this is the formation of a transcription terminator. If tryptophan is not present, then the Trp sequence cannot be properly translated and no terminator will be formed. So this way, premature termination of transcription will further decrease gene expression. And again, if there's no transcription termination, then transcription can proceed and the rest of the genes will be transcribed. In this example, we had two different ways of regulating gene expression that both responded to the same environmental cue. The advantage of having multiple layers of this type of regulation is again to prevent or even further decrease any leaky expression you might get for the repressor not always binding. Again, one simple way of thinking about this is that if you only had the repressor, you only had the anti-terminator form of gene expression regulation, you would have a gradual decrease in the amount of transcription or gene expression at this operon in response to increasing amounts of tryptophan. By having both, the transition between expressing these genes and not expressing them at a high level becomes much more steep. Transcription initiation and transcription termination are not the only two steps in gene expression that can be regulated. There are actually many opportunities to regulate the expression of a gene, and in fact most of the different steps involved in gene expression can be modulated or regulated by different factors in the cell and can respond or change in response to different environmental signals. This includes how much transcription actually begins, or how much translation actually initiates on an mRNA. How fast does the process occur? When and where do you stop, and how often do you stop or terminate each process? And together, this allows for multiple switches so that a cell can appropriately fine-tune not only the amount of protein, but also how it responds to different conditions and how fast it's made. There are even more examples of post-transcriptional control, and I'll show one briefly. These include mRNA degradation, so if the mRNA is degraded or destroyed, it will not be expressed. If the protein is not properly folded, it won't be active. If the protein is modified, it may either gain activity or lose activity. As we saw in the previous example, the phosphorylation of the transcription factor was necessary for its proper activity. And again, if the protein is degraded, of course, it cannot carry out its function. Here's an example of some post-transcriptional control. Here we have an example we saw before, but in this case, one gene is being expressed and translated into protein. The other gene is transcribed because RNA polymerase can recognize the promoter. But here, the mRNA is able to base pair upon itself and forms a secondary structure, and it can no longer be translated. There's no access, for example, for the ribosome to bind to the ribosome binding site or the start codon and begin translation. So this would be the case for this particular gene, no expression, at a low temperature or moderate temperature like 30 degrees Celsius. If you raise the temperature, whenever you raise temperature of any solution or any living thing, molecules will vibrate and move more rapidly, and so a higher temperature might not permit this same RNA folded structure to occur. The molecules and the bonds will simply be vibrating and moving too much, and the mRNA will unfold. So again, at this low temperature, we have a stable RNA structure, and we don't have translation. At a higher temperature, the RNA unfolds, and we're able to produce the protein. And in this particular case, the protein produced is actually sigma 32. So this is the actual way in which the production of sigma 32, the heat shock sigma factor, is regulated. Now, since sigma 32 is produced, this will alter which genes can be transcribed in the cell, as we saw before. Again, this occurs at a higher temperature, for example, 42 degrees Celsius. Another example of post-translational modification or post-transcriptional control is the MazF-MazE toxin-antitoxin system that is found in some bacteria. The MazF protein functions as an endoribonuclease, or protein that will recognize certain sequences in an RNA and cleave that RNA by hydrolysis, right? Rendering that RNA or that mRNA non-functional, or cannot no longer be translated by the ribosome. The MazE protein can bind to the MazF nuclease and inhibit its function. The key to this system is that the MazE inhibitor is somewhat unstable, and certain types of stress can lead to its destruction. And if MazE is no longer active, MazF has free rein to destroy RNAs. This can actually be used to help radically alter very quickly the gene expression in a cell. For example, on the left, we have transcription and translation of the MazE and MazF proteins, and together the two proteins will interact, and MazF will not be able to destroy any mRNAs in the cell. If there's a stressful condition on the cell, MazE will be destroyed, MazF will still be around, and it will be able to degrade many of the RNAs in the cell. Not all, depending on which sequence are present in the mRNAs. Again, this is going to radically alter the type of gene expression that is possible in that cell. So just to summarize, we've seen a couple different examples, specific examples, of how gene expression is regulated in prokaryotes. This included the use of sigma factors to recognize certain sets of promoters, the use of activators and repressors to control whether or not transcription occurs at, for example, the Lac Operon or the Trp Operon. And this included a lot of post-transcriptional and post-translational methods of regulating gene expression. Again, for example, protein or RNA folding to prevent translation, or protein folding or protein modification, which controls activity of a protein.